Hello guys, my name is Dr. Varun and here I am back with 30 days, 30 most important topic for NEET 2025 Biology.
Chalo, let's complete it.
Well, in today's videos, we're gonna talking about from the chapter Respiration in Plants, that is from plant physiology, and the topic that we're gonna be discussing is electron transport system. Well, where it is taking place? It is taking place inside the mitochondria. Where inside the mitochondria? Its complexes are present on the inner mitochondrial membrane. So if you know that mitochondria is looks like this. So if you see that this is what outer membrane, it is the inner membrane. The inner membrane is where all these reactions are taking place, and they are doing what? Oxidative phosphorylation, which means what? Phosphorylation means ATP formation, and oxidative means oxidation taking place over there to make the ATP. Now for oxidations to take place, we have to do something with this proton or electrons. Basic chemistry, if you have remember the redox chapter from there. Okay, so that concept is only coming up over here. So we have understood the basic where it is taking place, what we are doing. Now why we are doing it. So if for this I have to take you something over here. If you remember, this is the aerobic respiration, one of the point of the respiration in the aerobic organisms, where what happened, there are structures like this is being formed. Now these are what? These are the higher currencies of energy. Means, for example, they are the dollar, and the dollar does not work in our country. In our country what works? Rupees, rupiah, and that rupees are what? Actually the ATP which is working in our country. So cell also is like that. Cells work only with the ATP. It can't take the help of NADH and the FADH2. So it want to change it into ATP. And then what will happen? If you have a dollars, you will go to the bank, exchange banks, and exchange bank will change it and give you the rupee. Same over here, this NADH and the FADH2, it goes to the change bank, which is what? Inner mitochondrial membrane, and it will change it. So this is only we're gonna see that how they are changing up. So here two different higher currencies presents. One is NADH, another is FADH2. So this is the whole structure in elaborated way shown to you. So I am just enhancing it. So what is electron transport system? It is making the reduced energy ATP from these two components. So this will make different amount of this will make different amount of it, and their pattern is different where they are going and making the energy. Now over here, if you see that this is what? This is the outer mitochondrial membrane, this is the inner mitochondrial membrane. First thing, the next most question that will come to you, what are the complexes present on inner mitochondrial membrane? On the inner mitochondrial membrane, we have complex number 1, 2, 3, 4 and 5, and they have a name present to it. For example, complex number 1 name is NADH dehydrogenase. Like this, every of these complex has what? A name present to it. Okay, this is succinic dehydrogenase, this is cytochrome BC1 complex, this is cytochrome C oxidase complex, and this is FATPase synthetase complex. Okay, so this we have to remember. Next thing we have to remember that what are the inner pigments present into it, these things. Okay, so this thing also we have to remember over here. Okay, which we will do it one by one. Let's just see that what is happening else. So here the outer membrane is there, here the inner membrane there. In between them, this space is known as perimitochondrial space, where this is what? The matrix where the aerobic respiration cycles are going on, means the link reaction and the Krebs cycles taking place. Let's just come back. First we're going to be study about the how this is forming the energy, NADH. Now for finding this NADH, I want that certain thing should be hidden up. What should be hidden up? This complex should be hidden up, because this is not working over here. Okay, so complex number 2 is not in our thing. Okay, I will make it little bigger. Back. So this thing is not in our talk right now. Okay, so I have changed it. Let's just see that what is happening with NADH first. So NADH will broke down into NAD positive plus 2 hydrogen, 2 electrons. Lot of them will be broken down. Now this NADH which is being broken down over here, it will go back into the aerobic cycle and again being forming NADH during that aerobic cycle formation, TCA cycle formation. Whereas these two things will goes to the first complex. As it goes to the first complex, it will be received by FMN. This you have to remember. This FNN will do what? It will throw the electron to the next one, FES, but throws the hydrogen to the outside. How we will remember it? That think about that if you are giving someone something extra, which they don't want, they will give it. So they gave the positive charge outside, negative to someone else. So certain people are positive to someone, negative to someone. The positive charge, they send it what? Outside. Okay, whereas the negative charge goes to FES. FES said that I am not taking this, let's just give it. So it gave it to the ubiquinone, which is present outside the complex. It is present on the inner mitochondrial membrane, which will carry the electron, accept the electron and give it to the third complex. So second complex bypassed over here. This is very important that it is not working in this NADH. So this goes and give it to the third complex, and in that complex whom it is given to? Cytochrome B. Then the cytochrome B will taking the negative. The cytochrome be like, why I am taking negative? I want something positive to balance it. So what will happen? Then again the hydrogens will come out and goes to it. Okay, fine. This hydrogen goes into it. Meanwhile, from where this hydrogen is coming up? This hydrogen is coming up from here. So over here only not 2 H positive but 2 extra H positive. Another one will breaken up. Okay, they together 4 will go out. Okay, as the 4 reaches here, it goes and couple with the last complex, that is ATP synthetase, and it will come inside. So what happened? 4H positive again come inside. When it is coming inside, what happened? The ATP is formed during this side. So I have shown you that ATP formation takes place. Whereas this 4H positive which comes inside is being taken over here. The question is answered. Now what happened? This cytochrome B which has 4H positive which we has taken in compensation of the electrons, it give it to FES and then FES given to cytochrome C1. Meanwhile, cytochrome C1 will throw the 4 hydrogen outside and 2 electron it will give to cytochrome C, which is also a mobile carrier which will take from this complex to the next complex, that is complex number 4. What is happening with this 4 hydrogen which went out? It again come inside. And as it comes inside, it forms what? Again ATP. Second ATP is being formed. But 4H positive are still here. So 4H positive over here stays. Whereas what happened? That 2 electrons which are present here, it goes to the 4th complex, first pigment present inside it, that is copper A. Then it goes to copper, then it goes to cytochrome A. Then it goes to cytochrome A3. Then copper B. Now meanwhile, this copper B also take the hydrogen from here. This hydrogen will be thrown out, whereas this electron will be given inside the matrix, and this electron will come back again and forming one another ATP over here. Okay, meanwhile what is happening, that the hydrogen which came inside, okay, which has been taken here, it came inside. That hydrogen also go there. Hydrogens join with the electrons. Electrons join with the oxygen, because all of this reaction is taken place by the oxygen only. So they join together and form what? Water. Okay. So here what actually we have seen and understand. We have seen that purposefully we are throwing the hydrogen ions outside. And always remember beta, this thing again will come in chemoosmotic hypothesis, that when from one side to the other side we are throwing things and its concentration is increasing and it wants to go back to its normal site, it requires energy, and ATP synthetase enzyme, when will throw the things back, simultaneously it will make the ATP. Okay, so how I will remember it? What we are doing with the hydrogen? We are throwing hydrogen outside. Hydrogen is what? or the protons is what? They're highly angry that how can you throw me? How can you throw me outside? I am not some kind of a basic element. I also have some power. So we're like, sir, what power? Huh? Tell, what power? So the hydrogen will say that, okay, hydrogen goes to ATPase and it goes inside. As it goes inside, all his whatever the aggression change into what? ATP. It makes the ATP. Meanwhile, it come back inside. So like, you saw na, finally I have did something na. So it makes the ATP. So remember it like that, that wherever from higher concentration hydrogen will be accumulated by this electron complexes, it will do what? When it will come inside, so inside only it will make what? ATP. Here remember this point, this is what which is embedded to the membrane. This part is F0 particle of the complex, whereas this is F1 particle of it. So F0 particle is integral part of the membrane. F0 is present towards the matrix and ATP will be forming inside the matrix. This was the one thing. Let's just see that what is happening with FADH2. In FADH2, what happened? Your complex number 1, that is NADH dehydrogenase, name itself saying it will not work because that is not working over here. So we will just mask it. So 1 is not working over here. Okay, if 1 is not working where, everything start from where? Everything start from the first complex. So here again it will broke down, it will accumulate 4H positive and it will be given to over here. So over here if you see that first it goes to FAD, then it goes to FES and then it will be given to ubiquinone. From ubiquinone the electrons will go over here. Okay, but hydrogen which has been broken over here, it will enter directly here. Nothing will enter over here. So this is cytochrome B, which was like, I am not taking the electron unless there is positive charge. So by okay, let's give you positive charge. So we gave the positive charge over here by accumulating 2H positive over here. It will goes to FES same, it will goes to this and the 4 hydrogen will be thrown out, electron will be given to this. This hydrogen will come inside forming 1 ATP. First ATP is formed by what? Third complex in the FADH2. And then the 2 electrons goes to this whole same thing over here. Simultaneously the 4 hydrogen which came inside will go again over here. This will throw the electron inside the matrix, which will be taken up by the oxygen and hydrogen outside. Hydrogen again will come inside, finally joined with this whole complex, again forming water. So look over here, how many times hydrogen come inside? 1 time, 2 times, because this blue color line is not forming. So if 2 times is coming means 2 ATP is being only formed. This blue color ATP won't be formed by the FADH2. I hope you are able to understand. It's a quick thing. Read again, go through again, see this diagram, what was happening, and you will find this notes in your group also, that is Biology Simplified English on WhatsApp channel. You can find it out. Link will also be given to you in the description for my channel. And that's all. If you didn't able to understand, do let me know, that I will try to explain this thing from NCERT line by line also. But after reading this, NCERT is just made the things little up, bundle up together. Means they are telling about NADH and then FADH2 also simultaneously. But remember that since I have told you now that from here the things will be common. So that's why they have coupled up together. Okay, the only thing you have to remember is that in NADH2 system 2 is not working and in FADH2 system 1 is not working, means complex 1 is not working. Well, I hope you have understood this topic. If you like this topic, give it a like, subscribe if you haven't subscribed, share it with your friends, or maybe don't, be selfish. And well, that's all for this video. Thank you.